Hello and welcome to this session of ACS Science Talks, Connecting the World Through Science. This is the virtual lecture series, Scientific Talks by specialists on specialized topics for a specialized audience. I'm Kunal Gupta and I'm pleased to be your host for today's broadcast of ACS Science Talks. Now, before we begin the session, it is my pleasure to invite Dr. Diksha Gupta, Director, Global Strategy for Society Programs, for an opening remark. Diksha, over to you.
Thank you, Kunal. I'm pleased to welcome all of you to this broadcast of ACS Science Talks, a platform by ACS to connect the global scientific community. As the Director of Global Strategy for Society Programs at ACS, I'm honored to interact with you today and share our global outlook and mission to support all science professionals. At ACS, we are dedicated to provide the necessary resources for students, scientists, academicians, industry professionals, and broader scientific community to thrive in the dynamic world. Our focus is on fostering innovation and effectiveness in scientific education. We provide access to opportunities, resources, skills training, and networks, ensuring that you have the support you need to excel in your endeavors. By doing so, we contribute to the growth and success of the global scientific enterprise. We believe that diversity, equity, inclusion, and respect are fundamental values that drive progress. We are committed to promoting these principles as we work to identify and dismantle barriers to success. Our aim is to create a welcoming and supportive environment for all individuals, regardless of their background or circumstances. Together, we have the power to bring a positive change. Through collaboration, knowledge sharing, and the application of scientific expertise, we can address the most pressing challenges being faced by humanity. By supporting science professionals like yourselves, we are laying the foundation for a brighter and more sustainable future. I'm grateful to each and every one of you for your dedication to the scientific enterprise. Your work is essential, and it's our privilege to stand alongside you in this remarkable journey. Thank you. And I'm sure you will have a great time listening, Professor Walker. Over to you, Kunal.
Thank you so much, Diksha. That was a wonderful remark, and thank you for joining us in today's session. Our speaker for today is Professor Gilbert Walker. Gilbert Walker is a distinguished professor at University of Toronto and the editor-in-chief of the ACS journal Langmuir. His surface science group develops and applies nanoscale technologies for characterizing biophysical interfaces and photonic materials. Walker's fundamental studies of polymer and biomembrane mechanics have recently led to platforms for super resolution imaging, biomedical diagnostics, and drug delivery. He helps to lead the NCE, Nanomedicine Innovation Network, which helps more discoveries from university labs to clinical applications. A commercial fisherman in his youth, he stays connected with that community by innovating materials for fish farming. Thank you, Professor Walker, for joining us. The stage is yours.
Thank you very much. Thank you very much, Dr. Diksha Gupta and Dr. Kunal Gupta. Thank you very much for your introductions. I really appreciate it. I'm very thrilled to be here today. I have a long-standing love for India and have my closest collaborator over the years has actually been a gentleman at TIFR, Dr. Sudipta Maiti, and this is a long-standing and deep connection that I have. And I was very fortunate to spend a sabbatical period with him actually several years ago. So I'm very glad to be here, and I hope my seminar will show some of the connections that I have that are quite strong to Indian culture and to the science that stems from that culture. And so in addition, I would like to make a small advertisement for the journal that I happen to represent. Langmuir is a surface science journal, and we're very, very excited about it. We're very excited about our ability to bring both fundamental and applications to its audience, who are people like you. And we're very much looking forward to the opportunity to see papers that come from you, manuscripts that come from you. I should point out that, in fact, Our authorship from India is really growing, and we're very excited about that. We want to increase this, and I want to comment that we have programs not just for senior authors, but also for more junior researchers and faculty. And I eagerly encourage you to take a look at some of those opportunities to interact. Langmuir is a great journal, and we have had a strong tradition in surface science going back now about 25 years. What I'd like to tell you about is a series of stories that are related to the marine environment. As Dr. Kunal Gupta pointed out, I do in fact come from a fishing background or a marine background. That was my earliest serious job. And for several years, I was a commercial fisherman during the summers, not while I was studying, but it's how I made my money. And in that process, I learned a lot of things about how that community of people was changing. And then when I was in India, of course, I ate lots of fish from India, which was quite delicious. What you see on the screen here are a few images of the people whose work I will talk about today. These are three tales that I will present to you. And these three tales were really, in many ways, stimulated. The fact that I went on this journey was stimulated by the gentleman on the far left, Dr. Nikul Gunari, who is now, I guess, vice president for research at a large plastics company called Goware, which is in Pune. And in addition to that, there was work that I'll present that comes from Doctors Kim, Zia, and graduate student Kelsey Wan. We have a very diverse and very powerful group, and I'm very honored to work with them. So let me tell you about our first activity. The first activity, or the first chapter, is really about aquaculture. As mentioned, as a child, I worked as in the commercial fishery, and in this commercial fishery, we took a lot of pictures as we were doing work, or my mother did. And in fact, what you see here is a picture taken by my mother back in around 1991 or 2, I guess. And in that picture, it shows people catching lots and lots of fish. Where I grew up, there were lots of codfish being caught. I could catch 4 guys, and I could catch enough to fill a classroom from floor to ceiling in an evening. So that we could catch a lot of fish, and that was a big business. Unfortunately, that over-exhausted that environment, and there's now aquaculture. So many of the independent families who were fishermen at that time, and independent workers and had their business as fishermen, now actually work in the aquaculture industry, earning very small amounts of money. So I went to visit some of those friends of mine just when I went back home, and to see what they were doing. And they told me about some of the issues that they were facing. In the aquaculture system, we have a different environment than an open and wide environment. In the aquaculture system, there are these net cages, and you can see them on the screen here, that have fish inside them, which are very crowded. And then the fish are fed, and then after they grow up, they are harvested. So one challenge that happens when the fish are really concentrated like that in an environment is that they develop parasites. Another problem, and with these parasites, as you can see from these pictures, are quite detrimental to the fish health, and also to the fish quality of life. And so this is something that is not well controlled. And I would like to tell you about one of our activities that has tried to address this. What fish farmers do at the moment is they use technologies which don't work very well. Some of them are passive, and they're clever ideas. So, for example, you can introduce another kind of fish in the cage, which is a cleaner fish, which will eat these bugs off of the in this particular case salmon. But I should point out that the lice that I'm showing you here, which are present in fish farms in Canada, in Nova Scotia, in fact, in India, where in the western coast of India, where there is fish farming going on, Kerala region. In that region, There is also a fish farm, and the Caligula species of the lice. They're not that this genus of the lice are there as well, and it's also an issue. So one strategy that people use here in Canada has been to have cleaner fish. It hasn't worked very well. Then there are more shocking methods. Actually, a hot water bath, it turns out, can get the bugs off of the fish. And even really startling and scary things like there were examples of efforts to test using hydrogen peroxide, which, as you can imagine, is a terrible thing to pass a fish through as a solution of hydrogen peroxide. So the farmers do various things, but the main thing that they do actually is to use chemical compounds that manage to somehow impede the development of the lice. But those methods are getting overused. These pesticide methods are getting overused, and they don't work very well anymore. Why don't they work very well anymore? Well, let's just think about a situation where you might have an analogous situation. This is a leaf, 4 leaves, and what you can see is that we have a collection of different bugs on the leaf, and after we apply a pesticide, if the pesticide is preferential for one type of bug, in this case, let's say the white-colored bug, then the white-colored bugs will tend to get reduced in numbers, but the red-colored bugs, which are resistant to that particular pesticide, are not. So then, as subsequent generations of bugs are developing, what happens is that the resistant bugs become the dominant group, and the pesticides no longer work. So what I want to tell you about is, so one of the big challenges in this business is to create not just one pesticide. Actually, worldwide, there is one used, really, emamectin benzoate, and it doesn't work very well, but it is what is approved. So what is in what I'm going to describe for you today is an illustration of an alternative approach that actually seems to work reasonably well. When we were initially working in this project with Nikki Gunari, we were actually treating the nets. We treated the nets around the cage in order to reduce fouling, which is basically, plants and stuff that grows on the edge of the cage. And when it grows on the edge of the cage, the water can't flow very well through it. There's a low oxygen content, and so the fish weren't thriving very well. So we actually developed a method of treating the nets with something that was not copper, and it worked quite well. One of the things that we tried was actually an extract from neem seed, and in India the pharmaceutical tree, right, the tree of life, the neem tree. It actually, what we observed was that in cages where we had treated the net with, and these are large cages, they're as big as a basketball court, so they're very large cages. What we discovered was that there was an ability to control the number of lice inside this cage, and we traced it to a development of the lice from the fish to the edge of the cage, and then they would get birthed and sent back forward. Now, the active ingredients in this is called azadirachtin. Azadirachtin is a triterpenoid, and you can see it on the left-hand side. And it's one of many different components in neem oil that affect insect development. In this case, they affect the ecdysone receptor, and therefore inhibit. If you bring this into the bug and it eats the bug. Is interacting with it, what happens is that they can't molt. Insects, when they molt, they change their shells. And if you inhibit the changing of their shell, they won't grow, they won't develop. So basically it becomes a way to impede their growth. And so what we have here in the composition that would be used from neem oil, and maybe some of you have actually used this, it has a variety of beneficial aspects. There are actually several different ingredients in it, but the dominant one that's important in this work is really the azadirachtin. So what we did was we did a test where we took 18 different tanks, and we put 26 salmon in each tank, and they were fed different diets. And when they were fed different diets, we were able to find out that we were able to observe that the development of the lice on the fish was very much reduced. Of course, we had different types of stuff provided. The reason that we did this as a feeding process is because this is the kind of thing that the manufacturer or the farmer really wants. They want something that they can integrate into their process, and it's very simple for them to integrate something into the feed. The feed then is basically an oil that's on the outside of the pellets that the fish eat, and then that goes into the fish and goes to the skin and affects the bugs. The nice thing is that this azadirachtin disappears immediately after you start, after you finish feeding the fish, and it really works very well. Now, this kind of technology sounds really quite interesting and quite good in some sense, but you might ask, is it a specific way of controlling pesticides? If it affects the lice, would it affect something else? So, for example, what you see in this right-hand side of this picture is an organism that we might worry about being affected. This is a lobster. Lobsters are a common crustacean in the marine environment in Nova Scotia, where I used to work, and they might be negatively affected. So this actually has driven us to explore other techniques, and I could explain those to you in another day, but they're in an earlier stage. But this is a great example of trying to find something by looking at a natural product that has a beneficial effect. I should point out that actually this technology is being integrated commercially in a different not by me, but by a different partner. Okay, so I talked to you for a moment about at the end about lobsters. Lobsters have a shell, and the shell that the lobster has is chitin. That's the hard shell. Actually, about 25 years ago, I had a visit, and the visit, the visit was from Cuba, and I had an email, and it said, "Hello, my name is Castro, and I'd like to visit you." And I was very excited about this, but then I realized, now wait a minute, this is a little bit strange. Fidel Castro is no longer with us, and it said Dr. Castro, so I was kind of confused. But anyway, I accepted the meeting, and I was and indeed, Felito Castro came to visit me, but it actually turned out to be his son, who is a nuclear physicist, and he came to visit me to my office, and it was a very good conversation. To be honest with you, we talked about many things, but one of the things he said to me was, he said, one thing that we have a lot of in Cuba is shellfish, because we produce shellfish. We take their lobster, in this case a spiny lobster, we take the meat and we sell it outside, but we have mountains and mountains of leftover shells. What can we do with these shells? And we had a conversation about that. And in fact, the use of the polymers that come out of lobster shells has a pretty long tradition. And what we have taken on then is in that tradition. The chitosan that we use, which is derived from chitin by deacetylation, usually it could come from either crustaceans or from fungi, but nonetheless, it's a processed component of the shell. Now, we're going to use this in a particular medical application. It's actually the chitin that is here. If you notice all of these sugars are components that actually, in terms of biodistribution in, let's say, a physical body, they have a particular tendency. The particular tendency is for those polymers to end up in particular regions of the body, and I'm going to take advantage of that in this project. Targeting a nanomaterial to a particular region in the body is an important way to develop a therapeutic. If you have liver disease or cancer or other kinds of liver disease, it turns out that one of the early and very dangerous developments is the development of fibrosis, or the fibers, the hardening of the liver. And so what you can see here is that, where there may be some additional, this is actually common for people who drink too much, but there are other origins of this phenomenon. Fibrosis is one of the early stages, which is quite terrible. So it's quite important to detect this if you want to have better prognosis. And if you have liver cancer, you would actually like to be able to target the similar regions in order to provide a therapeutic. So what we've done is we've taken the chitosan that we obtained. We have then functionalized it so that it has the ability to carry a drug by integrating a hydrophobic part, which is this palmitoyl part on the left-hand side. And then we take those guys and we bring them together in sonication to make a micelle. So a micelle basically now has an oily center from the palmitoyl component, and it has, of course, a hydrophilic outside. And then it's possible for us to do various things. We can use the oily region to carry a drug. In the case of cancer, we've done that actually to deliver things like sorafenib to help treat cancer. In this particular case, we are just going to use it to demonstrate an imaging technology. So by connecting this technetium ion in, we can feed this to animals, and if our targeting approach is working well, we can then image the biodistribution of that fibrosis using these technetium labeled systems. So here's kind of a little bit clearer picture. We start with the glycol chitosan, we do the palmitylation, then we make the micelle. And actually, I don't have time to show you the in vitro data. I took that slide out just a moment ago. But essentially, it does in fact deliver specifically to the fibrotic tissue, and this is a good way to make something that takes advantage of this particular material, which is naturally derived, the chitosan that can come from the shells. Okay, another material that is common from the marine environment. I used to collect this algae as a boy, bags and bags of it. And I think I got something like 5 cents per pound. And if you think about that's when it's dried. So if you think about it, that's a lot. So you might get, I don't know, $1 for a pretty good sized bag. So there wasn't very much money I made from it. And I used to think, what's better? What could I do better with this than just, get $1 or $2 for several hours of work? And in fact, there is a logical use of these brown algae. These alginates can be used as hosts for growing cells. And I told you a moment ago that we use the chitosan material for targeting particular tissue in rabbits in that particular case. We also are interested in using this hydrogel, which is again a polysaccharide-based material. In this case, what we're going to use it for is to create a host system. The host system is going to be for cells that we want to study. So why would we want to create a host to study a bunch of cells? Well, because I also am trying to develop drugs. And by making cells, in this case, cells of different types, and by putting them into a host, I can see how the drug develops and affects those cells. And in our case, because we're going to make very, very small hydrogels, they're going to be about 50 microns across, we can use a very high throughput method for analyzing them. So now we're going to be able to do that. And what the work will have is it will have two types of cells. One cell is the target cell, which is a cancer cell. And the second type of cell is a macrophage. That's a cell that can eat the target cell. And we want to understand how to turn on this process, because that's a way to treat cancer, is to turn on this immune response. Now, if you were to do this in a regular laboratory, and you could do this yourself in your own lab, basically what you need is a couple of beakers and a screen which is very fine, and you could do co-culture. You could put, one type of cell on the top, and another type on the cell, and you could separate those two cell types. And you could look at the interactions between the two, but it's relatively big, that object. It's the size of a tiny beaker. And so that's something that, and then if you were to analyze what's going on in that system, you would actually see the interaction of many, many cells at once, which may not be very good if what you're trying to understand is rare events. And in cancer, the rare events that may be important are related to the cells which may be rare in number initially, the ones that are cancerous. It's a little bit analogous to the case where we had some bugs that were resistant to the pesticide, right? Those are the ones that we actually want to pay attention to. So in our case, what we're going to do is, instead of using this thing, which is macroscopic, which is, the size, at least sort of the size of your finger or something like that, typically bigger, but something like that, we're going to now make it just a gel, and it's going to have the two cell types in there. They're going to be, cancer cells and macrophages, the eating ones. And what we want to do is we want to be able to create an environment that will have mimic the environment within which the cells are developing, and see if we can understand the effect of that environment. This is because in the case of the parent disease that's associated with these cell types, leukemia. In leukemia, the reason that many of the therapeutics that we have don't work very well is that the cells that are so terrible that lead to relapse or the recurrence of the disease, they hide inside, for example, the bone marrow, and it's very hard to get the medicine to those cells. So what we're trying to do is to develop a technology that allows us to look at rare and at small environments that we can modify and screen for heterogeneity among the responses of the cells. So this is what we're doing. It's a microgel screening system. Of course, we want to be able to make lots of the gels. We want to be able to put in different cell types. We want to have a healthy environment for the cell, and we want to be able to deliver drugs. So those are components that I need to show to you. We do this using microfluidics. Everything here is home built. We build it in our lab. And it's you can see the microfluidic system. There are two inlets, and in the two inlets, we bring in cells of different types. They are then joined. And what we do is we use, because the alginate has a negative charge, we use a divalent ion, in this case calcium, to cross-link that gel and to make something that is a little bit more rigid. It's more rigid because it will now more naturally mimic the living system that we would hope for the cells, and also, of course, it's easier to create something that we can study. So we basically make something that is, sort of a few kilopascals in stiffness. We can make these at high throughput. We can make millions of these, no problem. And the cells that we can put into these can actually then be put in different numbers of each type of cell into the microgels. So here is a flow cytometry image. And what's a flow cytometer? A flow cytometer is an instrument that allows you to take a quick snapshot in 1 millisecond of an individual cell or an individual microgel, and then you can use a fluorescent label that would stick to the surface of these cells to tell what kind of cell type you have in there. So it's basically a counting method of cells that have different labels on them. And what you can see is that we can get microgels that contain just the macrophage, or microgels that contain just the cancer cells, and then we can get the ones where we have the two different cell types in just one microgel. And we can image them directly. So here's what the fluorescence image of those guys looks like. So we can count them. We can change the number of the cells in each of these different microgel by changing the input concentrations of the different cell flows. But what's nice about this is we can do this with thousands and thousands of microgels to get good statistics. So what you can see is that we can preferentially get single pairs of cells of two different types in the microgel, or small numbers. Now, I had to show you a few examples that this system works pretty well. So let me make sure that background information is available there. And what you see is that the cells are proliferating very well. So that means the alginate is working for us. Just as a point of reference, it is necessary to purify the alginate quite well. So that's good. We know how to do that. The next thing that we want to do is to figure out whether we can get our drugs to go into the alginates. And then we want to watch, how the cells respond. We want to see whether the cell, what happens in their interaction. And we want to look at the different sort of proteins that are given off because they teach us what's happening for those cells. So the way we deliver our drug is by using lipid nanoparticles. So the lipid nanoparticles that we use are related to the ones that are used in many vaccines. They aren't exactly the ones that are used in the vaccines, but they're related. And they're kind of small. They're about 80 nanometers across. They contain a few different lipids. And these are used actually to contain the nucleic acid. We're going to use, in this case, a marker of green fluorescent protein, but we use lots of different things. So this is basically a carrier to carry a drug example. It has on it a polyethylene glycol. It has in it something that can be used to condense the nucleic acid. And it has other things that help to affect the stiffness of the wall of the lipid to affect how that is taken up into the cells and how they are released. The drug is then released once you get into the cell. By the way, this is a very inefficient process. There is a lot of value to studying to do a better job with this. So anybody who's listening to this should realize that only 1% of these lipid nanoparticles that are getting into the cell are actually then doing a job inside the cell. 99% of them are not working. So if there's an opportunity to develop something new, this is a great place. Because, why is 99% getting thrown away? So the reason that we have the polyethylene glycol on the outside is because the polyethylene glycol helps to keep things well solubilized. It also helps to prevent things from sticking to the outside of the lipid so that it doesn't get, opsonized and removed from the bloodstream so rapidly. That's the reason that we have this kind of thing on here. It's a very common solution. And it creates if you were doing a physiological system, it would give you long circulation that would stay in the bloodstream for a long time. But there's a big disadvantage that many people aren't aware of. That is actually, this use of PEG, which is actually standard in the gene therapies that exist in the world. That polyethylene glycol can be used once. Why? Because your body actually will develop an immune response very rapidly. Your body has the ability to recognize PEG, polyethylene glycol, and it will develop antibodies. So the second time that therapeutic is delivered, it won't work very well because the body will phagocytose it, will throw it away. So basically, this is another big challenge. All of the technologies that are commercial for the lipid nanoparticles are PEG-based. They don't have a lot of value as repeat therapeutic. And as you know, if you want to treat somebody's cancer, it's rare to have a single treatment. Okay, so let's take a quick look to see how this delivery worked in our system. And what we see is that the lipid nanoparticle is uptaken, and we see that also we can get good expression of the messenger RNA to produce the green fluorescent protein. So that's good. We've shown that we can deliver this this way. Other things that are worth noticing, we can use this technology to identify what type of macrophage we have. There are different types of macrophages. There are macrophages that are of different types. Some are inflammatory, some are not inflammatory. They have different subphenotypes even within that. So we can measure the markers for those. We can measure markers on the cancer cells as well. These two markers, calreticulin, CD47, they represent two markers. The CRT or the calreticulin is an eat me marker. It is a cell saying to the outside that the macrophage will recognize as something to induce phagocytosis or for it to start eating that cancer cell. On the opposite side, CD47 is a don't eat me signal. When there's this bloom of the CD47 on the surface of the cancer cell, then the macrophage can't capture or avoids that cell because it goes to, it's kind of a marker of self, and it doesn't want to destroy that. Okay, the bottom line is that we can in fact evaluate phagocytosis, which is the, in this case, the immune-driven consumption of the cancer cell by the macrophage, and we can also analyze apoptosis, which is basically cell suicide. We can identify which is which. And we can also measure all the markers that are relevant. These are the immune markers that tell us how the macrophage is functioning. So this is important if we want to study the immune system, to be able to analyze these different types of markers. So I've shown you that we're able to grow the cells nicely. We can do them in lots of gels. We can deliver drugs, and we can analyze how well those cells are functioning. I have a very small amount of time left, and I want to show you with one quite interesting figure, or at least interesting from my point of view. Now if you remember from this initial device, when we created the microgel, we used this microfluidic tool, and we had two different inlets, and the two different inlets were coming together to produce something. It's probably hard to see on the screen. What you see are sort of two hemispheres that are coming together. To create the object, which will be the final microgel. And the cross-linking gets them to stick together, the final cross-linking. Now, what I showed you in the initial data was where we used always the same kind of alginate. But we had an idea. Our goal is really to study microenvironments. For example, how the cells that might be stuck in bone marrow or other environments where there are more hydrophilic regions, more hydrophobic regions, stiffer regions, softer regions, how this differential environment could affect the interaction between the macrophage and the target cell. So what we did is the following. We basically made an alginate that is either pure alginate or alginate plus gelma. Gelma is a collagen-based polymer that it contains a collagen-based polymer that gives an additional way to cross-link, and also gives a way for the macrophage to grab on and pull itself around inside the microgel. If you will, the cells will have different abilities to migrate in the alginate as compared to the alginate plus gelma. Okay? So what I want to tell you, I don't want to make you go through all the data, but the bottom line is this, and you could tell it, if you take a screen snap, you'll be able to check for yourself this later on. What we have discovered is that by doing an experiment where we made 4 different types of gels, okay, with microgels. One is both alginate, one is alginate plus gelma, one is we've changed to be gelma and alginate on the right side, and then the other one is both gelma and alginate, and then we change the cell type that's in each different compartment. What we discover is the following. We discover that the initial environment that the macrophage is in influences its ability to move into the second environment, right? To kill the second cell, it has to move from one half over to the other cell. So if it starts in alginate, it has a better ability to go over to another alginate half of the microgel. On the other hand, if it starts in the alginate gelma system, it has a reduced ability to go over into the pure alginate. And if you start in alginate gelma, then in both sides, then it efficiently goes across. So I think this is the first example of a high throughput system that can analyze that migration and phagocytosis capability in this high throughput way. So the conclusion really is that the local gel type modulates the macrophage motion and then the phagocytosis. So we're very excited about this. We think that we can use this now to teach us how to control immunogenic cell death. And I hope that someday I may be able to tell you about that. I want to acknowledge the students who have been working in this project. They are circled there with the red circles. And I'm extremely lucky to have been able to work with them. I keep my life keeps contact with farming, but it's not in the ocean anymore. But you can see that I help on a farm, which is in this case it's in my region, and my group research group goes out there periodically. This is actually a very good way for my research group to do something other than just working in the lab. We do stuff together. We work. We get exercise, and then we eat some nice food. Okay, so thank you very much for listening. I very much appreciate it, and I'll be very glad to take any questions that people might have.